Harold, it's an absolute pleasure to welcome you back to the GA4GH Plenary. You don't need much introduction, but for the people who do not know Harold, Nobel laureate, director of NIH, director of the National Cancer Institute, but perhaps most importantly, one of the true visionaries for open science over many, many decades. So Harold, we're very honored for you to provide a plenary talk, and over to you.
Okay, well, thank you very much. I'm getting a lot of feedback. Is it feeding back there as well?
It is.
So
I'm not sure. Maybe turn the volume down. Yeah.
I don't think that's enough. I think I'm on multiple, and I'm sorry for these technical glitches. They're beyond my comprehension. Ewan, thank you for hosting me. Very nice to be in touch with all of you. As Ewan knows, I've been involved with the GA4GH before it was called GA4GH, but it was simply an idea that many of us had that we ought to do something to improve the standards and the acceptance of genomics for many purposes, especially for health. And I've enjoyed watching the growth of this organization and the way it's become the global convener and the arbiter in the practice of genomics, with emphasis on standards for practice, ethics, mechanisms of use, communications applied to, often mainly to research, but also to clinical care and many other activities. This is a time in the history of this field when, for a variety of reasons, it's important to give further attention, additional attention to the question of the equity with which we practice genomics. And although I do have a role here as the chair of the Senior Advisory Committee for the GA4GH, I'm going to ignore that role and focus entirely on two projects that I've been involved with over the last year or two to try to improve the way we apply genomics to populations that have not been experiencing the benefits of it over the last several years. I can have the next, I can't control my own slides, so I've got to use the word next. So two examples I'm going to delve into a bit. First, the experience I've had over the last few months in trying to provide advice to the World Health Organization on expanding the application of genomics to further the goals of the WHO in global health. And I will tell you about a report that a new science committee is putting together, and how the GA4GH is and can continue to provide advice to us in doing this. Secondly, I want to speak more briefly to the development of a research consortium through the New York Genome Center in New York City to study the role of genetic ancestry in various aspects of cancer, including cancer risk, prevention of cancer, cancer initiation and progression, cancer outcomes, and so forth. And I'll come to that in a few moments. Next slide, please. So about 3 or 4 months ago, the World Health Organization appointed a new council to oversee the scientific activities that are connected to the goals of WHO. This slide simply illustrates the members of this small council. We only have 9 members, but there's extreme diversity, as you can see by looking at the location of the 8 other members from Asia, Africa, Middle East, Europe, and this group, which has also quite a lot of professional diversity, people who represent public health, who do research of various kinds from the clinical to the basic, have been brought together to, next slide please, to provide, next please, thank you, to provide advice to Dr. Tedros, who's the Director General of WHO, in conjunction with our close working relationship with Soumya Swaminathan, who's the Chief Scientist at WHO, to tell Tedros about new opportunities for applying recently developed science and technology to improve global health, especially in low and middle income countries. This council has begun to meet on a very regular basis. And we decided, without a whole lot of controversy, to focus our initial report on the various factors that are entailed in the wider use of genomics, especially in poor countries. Next slide, please. We tried to put out an initial statement to the world through a letter to Tedros and through some op-eds to announce that our council existed, that we have intentions to do a large-scale report. We couched this announcement in the context that everybody understands, the need to use genomics. I can have next slide, the next click, just click, please. Thank you. To use genomics in the current emergency context of the COVID-19 pandemic. And this is an example of an essay that I published in Financial Times last month to explain why it's important to be doing genomic analyses of viral pathogens, in this case, of course, SARS-CoV-2, and how the wider use of genomics just for this specific and highly understandable purpose forecast the need for broader uses of genomics, including human genomics, to deal with a variety of health problems that we have seen successfully confronted through the wider use of genomics, and to explain the rationale for expanding the use of genomics in even the poorest of countries. To take those steps toward this report, we have been setting up a planning process that, if I can, the next slide, please, has taken advantage of our friends at GA4GH, including Peter Goodhand, whom you'll hear from soon, and Ewan and others, to prepare for our report on genomics. And we've currently begun, well, more than begun, we've outlined the plans for 3 workshops that I want to describe very briefly and to explain how those of you who are affiliated with and engaged with the GA4GH can participate, at least indirectly, in these workshops that are intended to help inform our council to make the best recommendations possible for the implementation of genomics in the poorer countries of the world. The first workshop, if I can have next click, is addressed to the benefits of genomics. It will take place on November 5th. And the idea is to outline the reasons for making the effort to more broadly use genomics in all countries of the world, 192 members of WHO. And that includes most importantly, the use of genomics in medical care and public health, as illustrated by the pandemic, but by work in cancer and inherited disorders and metabolic diseases and many others. But we also want to illustrate and emphasize the other benefits of genomics for economic growth, for agricultural uses, which of course have implications for health through nutrition, through education in the educational domain, and many others. The next workshop is going to be conducted on November 19th, and it addresses the central issue that will be raised by virtually all governments and other organizations that would be responsible for establishing genomics in poor and low and middle income countries. And they include some of the technical and financial issues that have to be confronted to get genomics established, but then also we have to consider some of the issues required for the maintenance of an effective set of tools for doing genomics in these relatively poorly financed settings. That is, establishing and paying a cadre of skilled personnel, having equipment that's not simply purchased, but also maintained and serviced appropriately, flow of reagents at reasonable costs, governance system to ensure the fidelity of genomic methods, regulatory apparatus, and so forth. And we're looking especially for stories of experience with these impediments to implementation and ways that some institutions and countries have solved them. The third workshop will deal with many issues that are particularly well pursued within the activities of the GA4GH, namely some of the legal, ethical, and regulatory issues, ways in which those issues have historically been confronted in countries with more or less success, and some of the specific issues that will be dealt with in that workshop include data sharing, consent, equity, the risks to individuals who participate in studies or allow their clinical information to be used, the standards that have to be reliably set for genomics to be practiced with good effect in all countries, and how an approach to genomics intersects with cultural norms in a variety of geographical settings. The next piece of information tells you that these workshops will consist of formal talks, some case studies, open discussion, and these workshops will be accessible to the public through, and we will advertise in various ways how to get access. At the moment, we're very eager to have suggestions that we can use during the planning process, because these workshops have not yet been set in stone. And I provided an email address at sciencecouncil@who.international to allow you to send us any information you think might be useful and pertinent to the development of these workshops, as we and the representatives of GA4GH and of other organizations like H3 Africa and the G2MC and others help us in the planning process. Next slide, please. So I'd like to turn briefly now for a couple of minutes to tell you about another approach to sharing the benefits of genomics locally within our own population in the New York region through an initiative that we call Polyethnic 1000 that's being organized by the New York Genome Center, where, in full disclosure, I work part-time and many others do as well. The New York Genome Center, for those who don't know, is an organization established about a decade ago with the affiliation of about now nearly 20 institutions in the New York region, philanthropic support, some support from NIH and other funding organizations to do work that research and some clinical work mainly in three major areas: neurodegeneration, autism and other neurodevelopment disorders, and in cancer. And over the course of the last few years, my colleagues and I have developed this initiative, which is intent to supply the large databases that exist and are currently growing, but are populated largely with cancer genomic information that comes from patients of Western European descent, by focusing our activities on the ethnically diverse, underserved patient populations in the New York health system. So that includes African Americans, Hispanics, Asians of all kinds, South and Central Americans, and so forth. And as you'll hear, we've made some progress toward setting up this system. And I want to spend a couple of minutes telling you what's been entailed in just getting this research organization established, and what we're intending to do in the near and more distant future to try to promote a more equitable approach to cancer genomics that benefits people of all ethnicities. Next, please. So this slide illustrates in sort of kaleidoscopic color detail the ethnic diversity of New York City, and you can see Manhattan and the Bronx on your left. The lower right is Queens, Brooklyn, and some of the areas on Long Island. This is the region that we're focused on, although some of our participants come from New Jersey as well. And we have nearly 20 participating sites that are indicated by the little blue squares. These are largely hospitals and academic health centers scattered throughout the New York metropolitan region. We have at least 40 faculty collaborators. We've established a large number of working groups to deal with the regulatory and technical issues that are involved in this project. We've hired pathologists and site coordinators and have leapt through a number or leapt over a bunch of hurdles that have to do with approval from institutional review boards, settling legal issues and technology transfer issues between the institutions that are collaborating, and have identified scientific leads at each of these institutions to help us with this project. Next slide, please. So one of the first things we wanted to do was to show that the system actually worked. We could take samples from a wide number of hospitals, bring them to the New York Genome Center for ascertainment of pathology, and in the initial case, just a whole exome sequencing, although the goal is to do whole genome sequencing on the samples, as you'll hear, and to show this actually works. We can get some information from these samples, and a system with this complexity is actually workable. I'm sorry that the size of these objects on the slide is unfortunately much too small. I would scold any postdoc who made a slide like this, but what's shown on the left is the difference between self-declared ethnicity, as usually accompanies any patient record where people declare themselves as African American or white or Hispanic, where there are only 3 or 4 colors involved. Then by doing whole exome sequencing, we have enough information to begin using guidelines established by other investigators to identify the continent of origin, which immediately reveals that African Americans are partly of African origin, partly of European origin, or other major geographical areas. And the bottom panel on the left shows what happens when we look at more regional genetic indicators of ancestry. And although it's not really important to see the designations of all these colored blocks, it's clear that the ancestral complexity of these individuals becomes much more detailed than we could ever possibly imagine from any simple personal history and self-declaration of ethnicity, in a way that we think will be significant as we get deeper into this project. On the right is simply shown, but probably invisible, the kind of genetic evidence that we've gotten from looking at these samples. That is, many kinds of mutations, translocations, gene copy number variations, base substitutions from known tumor suppressor and proto-oncogenes, with lists of some of the tumor types that have been analyzed. And that doesn't require any more explanation. It simply shows that these analyses with samples that we've gotten from repositories in the participating hospitals do yield information that is relevant to and consistent with current practice in cancer genomics worldwide. Next slide. No, sorry, you missed one. Something's missing. Go back one. Back one. Can you go back one slide, please? No, there's something just missing. I don't know why. I sent it to you. Anyway, what I had on the slide that's missing is a description of the stage we're now in with Polyethnic 1000. We were able to accumulate resources, that is money, from a substantial number of donors and from some of the institutions that participate to set up a grants program in which we've funded 7 groups of investigators at many of these participating institutions to carry out 2-year projects that address 8 different types of cancer, largely in African American communities, but also there's one that addresses East Asians, and in the context of questions about possible relationships between some aspect of tumorigenesis or the response to therapy as seen in these ethnically diverse populations. These projects are funded with fairly modest resources, a few $100,000 each. They're overseen by senior scientists who work with us at the participating institutions. They're coordinated by two young faculty members, both happen to be African American, who are designated as our ethnicity and cancer scholars at the Genome Center. Those projects are simply in fairly early stages, but we are envisioning ways to expand our local network, to work with scientists in other cities, other urban areas who might be interested in setting up parallel efforts and benefiting from our experience. We hope to have a more expansive approach to ethnicity that is with many more projects addressing other populations than African Americans, with more tumor types than the 8 that are currently being addressed. To do this, of course, will require increased resources, and we're working with a number of funders to try to obtain those. The goal is to be doing whole genome sequencing and analysis, a special expertise of the New York Genome Center, and eventually to integrate that information with clinical and social information, working in addition more closely with some of the affected populations, using the skills of social scientists who can help with patient engagement, especially when there are cultural impediments to engagement of these individuals in prospective genomic studies. I'm going to conclude here and ask if there are any questions. I appreciate your attention to these 2 new initiatives, and I hope we'll hear from you during the question period and thereafter if you have suggestions about either the approach we should be taking with the World Health Organization to more widespread introduction of genomics into healthcare practices, and if you have suggestions about the work we're doing in the New York Genome Center on ethnicity and cancer, I'd be happy to hear about those as well. Thank you very much.
Okay, that's great. And Stephanie, if we can get the Slido up. Thank you so much, Harold, for the questions. Maybe as the questions come up, Harold, what do you think is the most important thing, with those WHO workshops? What's the most important input you need from the scientific community?
Right. Well, I think
yeah.
So I think the critical thing here, Ewan, is that we get the benefit of experience that people have had in trying to set up genomic enterprises in restricted settings. So I think we all are going to agree that there are many benefits to genomics, and that those benefits may vary from country to country, but for everyone there will be potential health benefits depending on the kinds of diseases that are prevalent. And there will be the prospect of kind of cultural benefits, social, economic, educational, that come with just an increased attention to modern technologies. But the place where I think things will be most difficult in trying to get a wider application of these advanced technologies will be in things as pedestrian as how reagents flow from the providers of reagents to countries, how service contracts can be established in an efficient way, how trained personnel can be, first of all, trained, and secondly, induced to do this work in a relatively limited setting rather than in the advanced economies where salaries are better and working conditions may seem easier. So I really urge people who are on this call who have had some experience in dealing with the establishment of genomic technologies in poor countries to let us know not only what works, but whether there are people who can speak to those issues. We haven't fully completed the invitation list, and we are looking for people who would be making useful contributions. Now, my goal is, as the chair of the council, is to deliver to Dr. Tedros not a 500-page compendium of what's great about genomics, but instead a short practical manual for how we can proceed to make parts of the world that, as you can see from this pandemic, often see the benefits of modern technology at the last possible stage.
Great. So that was so we've got some other questions. I noticed that Nicholas Rabin has actually answered one of the questions via this, which is great. But maybe this question, what about looking at pediatric diseases, including cancers, and about other omics? So I think this question here, Harold, is sort of how broad is your remit in WHO genomics? Is it, you know, where does this start and where does this end in terms of the sorts of diseases that you're looking at?
I mean, I don't think we're going to prescribe what, you know, there's so many countries involved in WHO, and the diseases that are prevalent in those countries vary a lot. So there are places where malaria is common, and the genomic methods that might be used for diagnosing malaria, identifying its strains, looking for drug resistance, those might be quite different from a country that has a high prevalence of some genetic abnormality that produces children with defects at birth. So I think the goal, from my point of view, is to establish a menu for developing genomic capabilities in a way that allows individual countries or institutions within countries to direct the use of their capabilities to specific problems that are particularly prevalent in that country. And I just think all we can do is illustrate why it's good to have genomics if you have an epidemic, why it's useful if you have a high prevalence of sickle cell disease of a certain type, why it's useful if you're dealing with the ancestries that produce diseases like Tay-Sachs or BRCA mutations. I think it's going to be up to the healthcare practitioners and public health experts in each country to decide how the technologies get used. Okay,
that's great. I think we've got time for maybe one more question, Harold, and just picking out an interesting one here. Are you going to look into the historical trace, the history of American ethnic groups via the genomes that you're getting in New York? And very practically, do you have a historian on the team?
Well, we welcome having one, and if somebody there who sounds like a historian is willing to volunteer to help us out, that would be great. You may have noticed that I did have the word history or historical on a couple of slides, and I do think that if you're going to understand the way in which people apprehend the use of genomics in the context of their own cultural traditions, which is something we face frequently in America because of our indigenous peoples, that it is important to know about the history of the ethnic groups that are being approached. And of course, you know, if you can't do this kind of work without collaboration with patients and with their families and with their communities. And we have been discussing with a number of social scientists in New York the best way to engage with patient groups and try to understand their perspective, because neither group, either the investigators who want to learn more, nor the participants who want to benefit from whatever is done without violating respect for their ethnicity and history, those things are going to work collaboratively to yield the best outcomes for everybody. It's going to be important to understand the history of the societies and the micro societies within our cultures to pursue things in the fairest and most equitable fashion.
That's great. Harold, I think I would certainly love to keep chatting, and that was a great slide about the diversity of New York. Very, very good luck with the WHO Council meetings. I know many people are going to come, and thank you so much.
Well, I just to say that we're very grateful to GA4GH, to you and to Peter and others for your contribution so far to this planning process. Thank you.
Right, with that, I think we go over to the CEO of Genomics England, not Genomics England, of GA4GH.
Thanks, Ewan. Can I have my